Hi, everyone. My name is Matt O'Donnell. I'm an account manager that handles the University of Kentucky. I've been with Joe for almost 13 years at this point, and we are so proud that the University of Kentucky subscribes to all Jove content. We are in more than 1,200 institutions worldwide. And right now on the app.jove.com website, as you can see. Jove is a scientific video resource that ultimately saves time, improves reproducibility, and accelerates discovery via visual peer-reviewed science. Jove has been around since around about 2006. A short little story and a little background about how the company started. The CEO, Moshe Pritzker, he was at Princeton as a graduate student, and he was trying to reproduce a stem cell growth technique using a paper. So his professor had him go after a specific paper, and he followed it to a T. He kept doing it and kept doing it over and over and over, but he just couldn't crack this specific stem cell growth technique. Luckily, his professor did have some grant money to get him a flight to go over to the original lab in Scotland to watch the scientists perform the experiment so that my CEO Moshe could learn it himself. After watching it in person, he was able to reproduce the experiment because he actually saw how they did it as opposed to reading text of the journal article. And that's how we came up with the idea of Jove. So that's what Jove is all about, science videos. We have more than 25,000 different videos across 13 journal sections, 6 encyclopedias, 18 Jove core video textbooks and growing, numerous science education collections, and 2 Jove lab manuals. And what we've found over time is that one video view can help a researcher reproduce an experiment. As I was talking about with our CEO Moshe above, seeing the experiment helped him reproduce it. And our Jove education videos are used by professors where they can assign this content to their students, and it gives them a visual basis for higher educational outcomes when they can assign this content. And to give folks an idea, I'll just get right into it and show you a snippet from a video article from the Jove medicine section, where it will really show you how the text of an article comes to life. And you may know some of these people, as this is a protocol from the University of Kentucky, which is really cool.
With this method, one can determine what components of platelets and the hemostatic system are important for thrombosis. Additionally, this method can determine how thrombus growth and architecture are affected at the cellular level.
The main advantage of this technique is that it allows a reproducible way to study various stages of thrombosis on ultrastructural level. A person new to this technique should avoid overhandling the tissue in the surgical area. This can lead to bleeding and can overcomplicate the identification of the carotid artery.
Begin marking the injury site after exposing the carotid artery by loosely encircling the region between 2 surgical threads having a 0.1 millimeter size. Place the probe under the artery, distal from the lower thread. Insert the plastic piece under the artery between the 2 threads to mark the site for iron chloride injury. Take flow measurements before performing injury to note the basal flow. Perform the injury by placing iron chloride soaked filter paper on the artery for 3 minutes. Remove the filter paper after 3 minutes to prevent the variable extent of the injury and to facilitate flow counting by the probe. Add saline at the injury site to remove any excess residual iron chloride. Monitor the flow reading. When the flow drops below 50% of the initial value, remove the probe. Quickly dry the area and add the fixative to fix the injury area externally. Next, promptly hold the artery near the injury area with forceps. Cut downstream of the lower thread and upstream of the upper thread. Put the tissue on the plastic tissue culture dish in the same orientation as it was collected and add a few drops of the fixative. Using a scalpel, clean the extra tissue around the artery. Ensure recording the orientation of the blood flow by cutting one end horizontally and the other tapering or oblique.
So the folks at the University of Kentucky did great work with that particular protocol. So reading from the text from this article and then reproducing what you saw here would have been much more difficult or maybe impossible. But we have a high quality video. And so we send videographers into labs to shoot the footage and then it's edited down to typically between 3 and 12-minute videos. But yeah, having the video is helping these researchers reproduce specific experiments. I'm not a scientist in any way, shape, or form, but if I were trying to put together, I don't know, a cabinet or something of the sort from IKEA, a video on how to do it. So that's kind of how I see it from my perspective, because I'm not a scientist. So Jove Medicine, where we were with that particular video, is one of 13 Jove journal sections. As you see here, it's behavior, biochemistry, bioengineering, biology, cancer research, chemistry, then bioengineering, environment, genetics, immunology and infection medicine, and neuroscience. There's also the Jove Encyclopedia of Experiments, which is where I'll take you now. There are 6 different encyclopedias here, and these are shorter portions of really important research experiments. And I'll quickly take you to one now to give you an idea.
Glucose, a primary cellular energy source, enters cells via cell membrane glucose transporter proteins. Glucose gets broken down in the cell cytoplasm to pyruvate via the glycolytic pathway, generating energy through adenosine triphosphate, or ATP, for cellular metabolism. To measure intracellular glucose uptake, begin with an adherent culture of fibroblasts in a multi-well plate. Replace the media with glucose-free media and incubate. The cells metabolize the available glucose to produce energy, attaining a glucose-starved state. Replace the media in one set of wells with glucose-free media containing insulin, a glucose uptake stimulator, and fluorescent deoxyglucose, a glucose analog. Treat another set of wells with glucose-free media containing fluorescent deoxyglucose. During incubation, insulin stimulates the translocation of additional glucose transporters to the plasma membrane, facilitating increased fluorescent deoxyglucose uptake in the absence of glucose. Deoxyglucose is phosphorylated and not metabolized further by cells. Concurrently, the extracellular concentration of fluorescent deoxyglucose decreases.
So as I was saying, the encyclopedia has biology, cancer research, biological techniques, immunology, neuroscience, and microbiology. And just like an encyclopedia, these are great reference points for scientists to go back to watch, shorter videos of important research experiments that are used often. So that's the gist of JOVE research. The second portion is JOVE education, and these are teaching videos. As I was saying earlier, there's 18 JOVE core video textbooks. These are much shorter video animations, and they can be mapped directly to textbooks. Like I was also saying, they're going to give students a visual basis for higher educational outcomes when faculty members are showing videos from some of these core components. They're not going to make up for the expertise of the brilliant scientists and faculty members here, but they're definitely going to improve student outcomes. That's what we found. So JOVE 4 is here. You can see biology, cell bio, molecular bio, A&P, pharmacology. There's some nursing content, medical, surgical nursing, chemistry, organic chemistry, physics, stats, mechanical engineering. And this content is growing as well. So I'll just jump in quickly to one of the A&P videos. These are really short, to the point. Interesting.
Human body structures can be organized into hierarchical levels. The chemical level is the most basic, where atoms combine to form molecules, such as proteins, lipids, and carbohydrates. Complex molecules further combine to form cells and organelles, specialized intracellular structures. Cells are the smallest living unit of the human body. They perform important biological functions, including synthesizing and breaking down biomolecules and communicating with other cells. Cells with similar structure and function are organized together to form tissues. All the different types of tissues perform distinct and specialized roles, such as muscle contraction or wound repair. Two or more tissue types together form the next level, the organs. Organs are special structures that accomplish complex functions. For example, the kidneys remove waste from the blood. A group of organs functioning together form an organ system, such as the digestive system. All organ systems collectively create a complete organism, the highest level of organization.
So these videos are often used for
Human body-
undergraduates, but they're also a great refresher for graduate students and medical students. And I'll quickly show a good one from the pharmacology. And as Joe grows, the videos get shorter and shorter, which is what I think young folks like nowadays, right?
Drug discovery is the process by which new drug candidates are identified to treat or prevent diseases. Drug discovery involves various stages: target identification, hits identification, lead generation and optimization, and drug candidate identification. First, molecular targets of the drugs, such as receptors, enzymes, and other functional proteins and nucleic acids, are identified using biochemical assays, genetic interactions, and computational methods. The next step is identifying hits, molecules that bind to the selected biological target. To do so, libraries of thousands of compounds, chemically synthesized or natural products, are screened using high-throughput assays. Hits that pass the primary screening are modified to generate leads that show pharmacological activity against the chosen target. These leads are optimized by combinatorial chemistry to
The next component within JOVE education after JOVE core is science education. And the science education videos are um, pre-lab, pre-lecture videos to give, like I was saying, students a visual basis for higher outcomes. And I'll just quickly jump around from with one physical examinations video where, like I was also saying, these videos, they're not going to make up for the experts that are teaching these specific things, but if a student can watch one of these videos before they get to the expert, it's going to really help move things along. So this is a palpation.
During a physical exam, the clinician uses the sense of touch through palpation to obtain useful diagnostic information. It is an assessment technique in which the examiner uses the surface of the fingers and hands to feel and examine an organ or body part. While palpation is fundamental to the diagnostic aspect of the physical exam, it is also important to acknowledge the role that touch plays in communicating caring and comfort during the patient encounter. This video will illustrate the different palpation techniques and discuss the approach and considerations for this procedure. First, let's discuss the different types of palpation techniques.
So what we're finding with these videos is that folks seem to jump to specific portions of the video, which is to the right here. If someone's struggling with the general approach, they can just jump to that specific part of the video and click play, and they're there, as opposed to having
Now
to
that
go
you
to
know
the
about
video
different
where
palpation
student can
techniques,
fast forward.
let's discuss the general approach to palpation during any clinical exam.
The videos can also be sped up.
Before every patient encounter, make sure that your fingernails are clean, groomed, and trimmed. Wash your hands with soap and water or apply topical disinfectant solution. Warm your hands either with warm water or by rubbing them together. If any specific infection control precautions are needed, then wear the protective equipment. And explain to the patient why it's necessary. Remember, gowns, gloves, and masks can present a barrier to building a relationship with the patient. In order to gauge the patient's comfort with the clinician's touch, you may start.
I think it's still somewhat helpful, even at 2 times, which is interesting. So science education, lot of great videos. They're a bit older, but there's a lot of great videos that are continually used year after year at institutions all over the world. The clinical skills content just gets used massively. The different section of clinical skills are physical exams 1, 2, and 3, emergency medicine, critical care, nursing skills. We have the COVID-19 procedures, physical exams 4. So all of these videos are just super important. We update them if there's something that needs updating. If a specific way of going about things is out of date, we will update those as well. And we hear feedback from institutions that tell us that we need to change this or need to change that. We can't do it right away, but we do our best to change these videos as quickly as possible. So all of this content, all of the JOVE education, JOVE research videos, everything can be embedded directly into faculty members' LMS. They can add videos to a playlist, create quizzes, and that's all. I'll just jump into one video here to show you where that is. Characteristics of life, cute bunny. Here's the embed video. You can add a video to a playlist, or you can create a quiz. We have curriculum specialists for each institution that will do all of this work for faculty members, and they're more than happy to help. But if you don't have interest in speaking with one of those curriculum specialists, you can do it yourself. So you just jump into embed the video. The instructions are here. And we'll jump to add to playlist. And just add, you can open up a specific playlist and just keep adding more and more videos. What I often tell faculty members is that, like, even if there's just one Jove video that can help a student, it's worth having that video for courses to send out to your students. But we're more than happy to help if you want a playlist of 10 videos or of 1,000 videos. We're more than happy to put those together. You can use whichever ones you'd like, of course. So like I was saying, the curriculum specialists, they can help with that. And they can walk faculty members through embedding specific videos, mapping them to textbooks. And I highly recommend, taking advantage of that service. And, even if there are 2 videos that you like the looks of, they'll make a difference in courses, but we're happy to set that up and do whatever folks need when it comes to playlists, quizzes, favorite videos, etc. And that's about it. So Jove Research, Jove Education, and there's also Jove Business. I don't know how much folks are going to be using Jove Business in the medical college, but they're fun videos for sure. Does anyone have any questions? Anyone in here have any questions about Jove content at all?
Could you show us the quizzes feature?
Yeah.
So,
So there's so many pre-made quizzes here already.
create your own business, as I saw on the video where it said create.
Yes, you can create your own quiz. We don't find that like a lot of folks are doing that. Because we have quizzes for yeah, for whatever you can do. So. And the quiz, these are newer, like our old quiz system is completely different than this one. So I'm not an expert on the quizzes, but I will share the information for the curriculum specialist that can walk those through that for sure.
So not every video has a quiz, but y'all are constantly making sort of make quizzes for video, so.
Yeah,
Yeah, gotcha.
yep.
I have a question. How do y'all solicit like updates to like protocols and techniques? Do y'all, do faculty submit? Can they submit to you like a journal, or do y'all do outreach to like get new ideas or get new updates? Like how does that work for you?
For Jove Research
Yeah,
protocols.
yeah.
Yeah, we have an editorial team that reaches out to folks to gauge their interest. And then if they agree to it, they submit, it gets peer reviewed. And the whole process, I think now with how quickly we're able to move things along with AI, I think the whole process now takes about 3 months or so from submission to the video being finished and edited. But it was cool to see Kentucky, so.
How often are the videos, like for nursing specifically, how often are they updated and content being created?
Yeah, so the clinical skills videos are updated on like a need to update basis if folks that are using them are giving us feedback as to things that we should change.
Okay.
But one of the newest collections is from medical surgical nursing. This is this is from, I think it's 6 months old or so. And the core nursing as well. So these are newer. We're finding that the clinical skills videos, so the older videos, are being used more, which is interesting. I know they're different. but they're being used more worldwide at the moment. So yeah, this is very new content. Yeah.